Semester once again. Do you guys have a good break? Caught up on your sleep? Staying up late, sleeping in late? That's kind of a good thing. So we'll jump right into our content though. We have an action-packed, a lot of labs this semester as well, just like the first semester. So we're going to be starting off with what's called serology. It's somewhat blood related. Serology deals with body fluids. It deals with biology, with antigens and antibody reactions. That's what serology is. So we'll start with that for a day or two, and then we're going to be going into the analysis of blood and blood spatter. It's a fun unit because you have a project where you're going to put together a video that could go a couple of different ways. You could make it documentary, or you could make it kind of like a crime scene and how you would recreate a crime scene with blood spatter. There's a lot of crime scene videos out there, like Dexter, to name one that really specializes in blood spatter. So you guys are going to be working on that project for about 2 weeks. It's got a lot of content in there.
Yeah.
Yeah, hi there. Yeah, you want to check it out? Thank you. Daniel here is going to check out our heat because it's a little cold in here, huh? About 66 degrees. Yeah. So the body heat's warming it up a little bit. So we are going to dive right into it here in just a few minutes, but just as a housekeeping reminders, if you have to make up any final exams, that's today and tomorrow specifically. That's why we have the schedule we do, where it's only going to like a half a day for you, but make sure you take care of business from the first semester if you need to do that. In this class, everyone's taken the semester exam, but if in other classes you need to make that up, don't make that a 0. So check with your teachers on that. And I sent out a progress report grade, which is, as far as I can tell, what your final exam, or I should say your final grade in this first semester is going to be. So grades are opened up to you on Friday from the registrar. That's when it becomes your official grade. There are a couple students in here that I think are settling for a particular grade. You can check that out from the grade that I've sent you, but your Aries is not available. That's why I sent out the grade report. So check that out. It should be in your email. And if you want to see notifications, remember that you can click over. Yeah, thanks. You can click into your notifications, which are, is it up here? Wasn't it a menu option? Maybe it's under settings and then notifications. Nope. Oh yeah, here on the website, it's under account up here and then notifications. So if you hadn't set that correctly, so that when things are graded and when I send out announcements, that it emails you. So you can have announcements emailed, or if you're on your app, it can then pop it right up on your screen. So stay informed. Now you guys, this is your last semester, so isn't it awesome that you are almost out of high school? For 99% of students, they're really excited. Others are a little nervous about the future. I think you guys probably have your college plans or what you're going to be doing after high school. So you're going to become completely independent. Even if you're 18 here at school, your parents can see your grades. But after this semester, it's you running and driving your own life in the direction that you want it to go. No one can see your grades. So how independent are you at taking care of business? If you've got A's and B's, I think you're right on track. If you have C's and D's, it's just maybe a little maturity that has to happen this last semester. I'm encouraging every one of you to do better the second semester than you did the first semester, it's like playing in a sport. You might be ahead at halftime, but are you going to just coast and go into prevent defense? You know what happens usually. You do worse in the second half of a game if you start relaxing and not trying as hard as before. So dig down deep and continue to get your A's and B's if that's what you're doing. Don't go into senioritis and then just let all your grades go. Do you know that if you have a college acceptance, they are expecting you to have the same type of grades your second semester? You have to give them your transcript. And if you tank your last semester, I've been around here to see the disappointment of a student losing their college acceptance because they let the last semester go. And now they don't qualify for the college acceptance that was having a prerequisite that you did your same average. So do that, or even do it better, because you want to go out of high school with a lot of pride, with a lot of determination that you can reach your goals. This is not a tough class. It's very easy to get an A in this class, a B in this class. Sometimes it takes work, I think, to get a D and an F. So don't skip out on doing the assignments. Get involved in the game. It's a lot more fun when you're involved. It's a lot more fun when you win. You guys know the Etch-a-Sketch? It's this little thing that has two dials. I was going to show you a picture. I didn't have time to pull it up. And you just sketch by dragging across all this like gray film, and it makes a pretty elaborate picture. Tip it up and shake it, and it's all gone, and it starts all over again. That's where you're at right now. Doesn't matter if you got a C or a D or a B. You all have an A plus right now. There's nothing in the grade book, which means that you have a perfect grade book. So now I encourage you on this assignment that we're doing today, make sure you do your best and you turn it in with an A. The next one, turn it in on time, do your best work, you'll get an A, and you'll ride that A all the way. It's so much easier to keep your A throughout the whole semester, but a strong start is really important. So I like teaching this class and the senior level, but I know that some are thinking, oh my gosh, one more semester and I'm out of here. But I'm encouraging you to really do a good job in all your classes. It really is something you'll look back on years to come, and you'll realize, yeah, I gave it my all. And that's what God expects of us, right? He's given us the intellect, He's given us the health, He's given us the opportunity to be here. So take full advantage of it. It really says to God, thank you for everything you've given me. Look, I've really done as best I can. I'm really proud of that. All right, we're going to get into our blood basic notes right now. And then in the next class on Friday, we're going to start class in here. Then we're going to go to the library and work on the computers on something called the blood typing game. It only works on a computer, so that's why we're going to go over there. I've already reserved the library. And then we're going to have a lab on our third class. Remember, in the first semester, you collected evidence in your evidence envelope, and the last thing we have is to determine if that's real blood on the cloth. You should have a sample of a bloody shirt from that. And so we're going to be looking at several different samples. And then we're going to get into blood pattern analysis, or reading the blood spatter at a crime scene. We're going to calculate the angle of impact of blood. You've seen this on television where they could draw lines with lasers or with strings, and they figure out where the blood originated from as it flew through the air. It hits different objects and then hits them at an angle, and you can calculate hundreds of angles of blood and then recreate a crime scene. So you're going to be doing some of that. That leads you into, as I mentioned before, this whole project, and that will take us into the middle of February. So after blood spatter, then we get into fingerprints, and we get into the analysis of bullets and trajectories, and also fire and explosions. And those labs are fun. No, we're not going to blow anything up or set anything on fire. We'll get some guest speakers in here. I hope to get firemen in here, also CSI for blood spatter analysis. And I'm going to work on getting the FBI in here. So it's going to be a fun semester. A lot of hands-on stuff. And we'll take that all the way to May. So please go to Canvas if you're not already there and grab the file that we're going to do today. It is called Blood Basic Notes and a Case Study. So we're going to watch this video right here. It is a forensic file where the blood drops. But before we get to that, we're going to do number one. We're going to go over some notes, fill in the blank notes. So go ahead and get those open in Notability. Oh, a word about being prompt too. We start the day here in block 3 every other day, so please be here on time. At least 6 of you walked in after the bell. So I didn't mark any tardies today. This is your grace period, but if you're not here when the bell rings, I will mark you tardy. You get so many tardies and you get assigned detention. So occasionally it's going to happen. That's why the dean's office gives you so many tardies, but don't make it a habit. In college, things are really serious. Like if you're tardy, they may not let you in. You might not be able to make up work if you missed it because you're tardy. And so attendance, it's really a measure of preparedness. So occasionally traffic or something like that is going to throw you off. But the people that walk in late on a day are usually the ones that make a habit out of it. So leave 10 minutes earlier, 15 minutes earlier, whatever it takes. It also reduces your stress because when you're trying to be on time, it's stressful if you know that it's not going to happen. So you just have to be a little bit more prepared. Wake up earlier, whatever it takes. So the blood basic notes here. Got the sheet? Here's the first one. What makes up the blood in our bodies? Well, on this page, you can see that there are things called erythrocytes, leukocytes, and we're going to talk about the plasma and the platelets. So fill in the blanks. The first missing one is called red blood cells. Those are actually called leukocytes. This is the part of our blood that makes it red because the red blood cells contain hemoglobin, which is the protein that is going to diffuse oxygen into it. And the red blood cells carry oxygen. It's the most abundant cell in our blood. It's not the only thing. There are many other things. The next main cell inside of our blood are called white blood cells. And the scientific name for those are leukocytes, but you have to fill in the blank. White blood cells. White blood cells are part of your immune system. There are, maybe if you took anatomy, you might know the exact number, but I think there's about 6 cells in our immune system. White blood cells being one of the main ones. And what they do is they're going to destroy viruses and bacteria that get into our system with cuts, with an infection that you might breathe in, that might be part of your food and get into your body. So however they get in, we have a fighting force. Your skin is your number one main defense system, but if you get a cut or a scratch, you know how it can get inflamed. It's putting blood over there and fluid over there to carry the nutrients that it needs to supply cells that need to grow to repair that damage. About half of your blood, it's just a watery mixture with the digested food in there. The amino acids, the building blocks of proteins, So the carrying system where things are suspended or dissolved into, that's called plasma. It's a yellowish liquid because of all the stuff that it contains. And electrolytes are ions that are part of our chemistry. You need to have potassium, you need sodium. These are all part of the cell process to make ATP and for your nerves to work. So again, if you take an anatomy, you really know what I'm referencing. And we're not going to go into detail other than know what makes up your blood here from this slide. And then platelets. These are the clotting factors. You know, some people have a genetic disorder where this platelet doesn't even get made. So if they get a paper cut, it never stops bleeding. They have to, with a paper cut, you don't really have to do anything. It's going to stop bleeding in a matter of seconds or minute. But if you are hemophiliac, you lack platelets, which it's almost like proteins that will just get in the way of the break and stop the blood flow so that you don't lose too much blood, and then the healing process can start. So it's the clotting factor, platelets. All right, let's move on to the blood facts. We have about 5 liters of blood. If you're smaller, you might have slightly less than 5. If you're larger, then you're going to have more blood. But 5 is the average. And that makes up about 8% of your body weight. So if you are 100 pounds, about 80 pounds is just the weight of your blood. So you see this on CSI shows. If someone, and we'll see it in the video today, if you get a wound, you can only lose about 2 liters of blood, and then your blood pressure is going to get so low because you've lost the volume needed to keep your heart beating. So certain wounds, if it is going to hit a main artery and too deep of a cut, then you could bleed out too quickly. So as we mentioned, blood is a living tissue. It has the red blood cells in it which carry oxygen, so that's why it's kind of interesting that blood is a living tissue. And we also have the white blood cells to fight infection, to heal wounds, so we've already talked about that. Letter C, to letter C. There are about 1 billion red blood cells in 2 to 3 drops. So we have an enormous number of red blood cells. We have somewhere in the neighborhood of like 10 trillion cells in our body. So we have billions and, like just huge numbers of red blood cells. Most of the red blood cells are made in the middle of your bones. Did you know that? In the bone marrow. So if someone has a blood disease, they try to go after the source of why they have a problem, and they can actually scrape out the marrow of their main bones, like in their femur, and then replace it with a healthy tissue from someone else, from a bone marrow donor. And that is then a pretty good chance that they're going to start growing that. Yeah, it's working now.
Did he come here or no?
Yeah, he did. Yeah. I haven't seen him since he walked through about 10 minutes ago.
Okay, and now it's working.
Yes, it is. Thanks. Yep, getting warmer in here now. That's nice. All right, moving on. Genetics of blood. Do you remember biology class, Mendelian genetics and Punnett squares? So you do inherit your blood type from both parents. Each of them gave you one of the genes. Are there? Okay, great. We're going over the blood basic notes, which you can get over at Canvas. Yep. Your blood type, just like your other genes, are established at the moment of conception. So before you're born, you're giving all your genes at the moment of fertilization. And you got one gene from mom and one gene from dad. And we're going to be talking more in detail about blood types on the next class and how it makes proteins stick out of the surface of your red blood cells. This is called antigens. Hey, aren't you familiar a little bit more now with like the spike protein of the COVID virus? The virus has a surface where proteins stick out of it like that. Those are called spike proteins for the virus. And those are antigens. That's similar to what all of our cells in our body have antigens sticking up like that, and they mark your cells as your own. Then your immune system should be able to recognize, hey, we just encountered this cell and we've never seen this protein before. It's probably an invader, and let's go after it and kill it. So it's like going after strangers inside your body. So when you have your immune system attacking a cell that it doesn't recognize the antigens, it will glue together. That word is called agglutination. So there are proteins that are part of your red blood cell that are going to attach onto the antigens. So you have an antigen-antibody reaction like a hand in a glove, and they don't let go. It's like a gang tackle in football. You see it all the time. The running back hits the line and he's got one guy on his one leg and then he's dragging, getting 5 more yards. Another guy grabs him. He gets another 2 yards. A third guy. So your immune system will tackle cells by attaching onto the antigens. And this is called agglutination when they're doing that. So here are the markers that I was referring to. If you have A blood type, you have A antigens. These proteins that stick out of the top of the red blood cell surface, going through the cell membrane. If you have B blood type, you have B antigens. If you have AB blood type, you have A and B antigens. And if you are O blood, you don't have these blood type markers. In parts of the world, they don't call it the O blood type, they call it the 0 blood type because you just lack the antigens. And then if you have A blood, you have antibodies, the white blood cells, that are going to be labeled as B, and they'll go after anything that's B. So if you have A blood type, you want to have a fighting force for everything else. If you are B blood type, you want to have a fighting force for what you don't have, so that it will recognize strangers. So you have A antibodies. So really, if you're A blood, you have the reverse of antibodies. So if you have AB blood, you can't have an immune system that will attack itself. So you can't have any antibodies for A or B, otherwise it would attack your own blood. And if you have O blood type, you have A and B antibodies. So we will work on that in the blood typing game in the next class. There are 3 alleles, A, B, and O. And remember, every gene comes in pairs because you got one from mom and one from dad. So you could get an A from mom and an A from dad, and so you are AA blood type. You could get an A from mom and a B from dad, and then you would be AB blood type. And if you got a B from mom and a B from dad, you are homozygous for B, meaning you have 2 of the same genes. So I'm talking some of the biology from a couple of years ago, and we're not going to go into much detail at all about the Punnett squares and stuff like that, but just as a reminder about the science with this. So that's what I was mentioning up here. These are the different blood types. If you're A, you could be AA or you could be AO. B blood type, BB or BO. O blood type, you can only be that. AB blood type, you can only be that. Now we're on number 5. How common are the 4 blood types? If you are A blood type, it's the second most common. You can say 39%. If you are B blood, you are in the 11% category of the population. Different parts of the world have different percentages. This is America. If you are AB blood type, 4% of the population. So the least common blood type is AB, and the most common blood type is O at 46%. So go ahead and write in those percentages. See this chart right here? Go ahead and fill in these letters because yours is missing it. So you're going to write an O at the top and an AB at the bottom. And it doesn't matter what order, but write A on the left and B on the right. This is the donor chart that you will be using on the blood typing game on Friday. So at the top, write universal donor. You don't have that word on the bottom right-hand side of your page. And you don't have universal recipient, so you have to write that in. The universal donor, meaning that this person can donate blood to any other blood type. O blood types can donate their blood to an A person, a B person, and an AB person. And then at the very bottom, if they're AB blood, they can receive blood from any person. That's why they're called the universal recipient. We're not taking into account the positive or negativeness of blood right now. We're just talking about the general letters. And then you can see that the arrows are already drawn in for you. So if you're A blood, who can you get a blood transfusion from? O. And always from the same that you have. So you can get blood from another A person or an O person. If you're B blood, you can only get blood from another B person or an O person. And AB blood, you can see that all arrows go to it. What can O get blood from? If they are losing blood, remember you have 5 liters of blood, and if they're losing 1 or 2 liters, they're going to need a blood transfusion. They can only get it from another O person. So that's how that works. There's also an RH factor. So apart from the antigens of A and B, there's also another one called the D antigen. And that's on the next page. It's called the RH factor. And they call it the RH factor because of the rhesus monkey. It turns out that apes and monkeys have the same blood types as humans do. And so they figured out this positive and negativeness of blood. It's another antigen. So apart from the letters that we were talking about, you can either also be an A or a positive or a negative blood type. So you can be A positive or A negative, B positive, B negative, A B positive, A B negative, O positive, O negative. And they figured out this negativeness and positiveness from the rhesus monkey, so it's called the RH factor, or sometimes called the D antigen. So it's another protein that's sticking out on top of the red blood cell. If you have it, you are positive. If you don't have the RH factor, you don't have that protein sticking out of the red blood cells. Now, if you were to get the wrong blood transfusion, if an A person gets blood from a B person, the blood is going to coagulate together and clump together. Remember doing the murder at Oldfields? and you were hitting the little circle. So that referenced that you had A antibodies, and you put the blood of the person in there. If it was A blood with A antibodies, they would clump together. And so the blood would then coagulate into like a gluey mess, and it would stop flowing through the blood vessels. So before like World War I, if people were given blood transfusion right there at the battlefield and they were just grabbing someone, they sometimes would get it right just by chance, sometimes get it wrong. If they got it wrong, they would kill the person. So someone figured this out, and they won a Nobel Prize for it around the beginning of the 1900s. And we're going to wrap up our notes here with blood being evidence at a crime scene. So as we saw in the murder at Oldfields, blood samples can be used to analyze suspects if they match the blood type. So if your suspect is A blood type and you have A blood at the crime scene, they might be a good suspect. If their blood types don't match, then they're not going to be a good suspect. But as you can tell, there's only so many, a handful of blood types, so it can only eliminate someone as a suspect, but it can't prove that that's the person. You have to do DNA analysis on top of that. So the blood droplets, which is called spatter, can be analyzed at a crime scene. It gives a lot of clues about what happened, how many blows occurred, what kind of object did they use. Was it a bat? Was it a knife? Was it a hammer? So they often can tell the type of weapon that was used based on the type of blood spatter that occurs, and so we'll go over that in the next few classes. You might want to circle or highlight that. Notice that it doesn't say splatter. There's no L in it. So when you guys do your movies, make sure you don't say splatter, that you say spatter. I know it sounds kind of like a technicality, but they're different. The words mean different things. So if you just get off on the right foot with how you say that, then you're going to sound more professional. So when you have blood drops at a crime scene, it's called spatter. Not like he went splat. Microscopic views of blood from different species. You can kind of zoom in on it. It's just the shape of the blood cells. But they all work similar. They have hemoglobin, which is going to diffuse the oxygen. That's the job of the red blood cells. So as you can see, there's another page of the notes, and that is for our case study. Oops, yeah, here we go. So we're going to watch this video right here. It's just 20 minutes long, and I think that's about enough time because that's about what we have left. 20, 25 minutes left in the class. Make a T-chart. It's already made for you, right? So if I open up your notes, the last page has the T-chart for you to write down. Oops, I lost it. There we go. There it is. This is a case where they're going to have the suspect, supposed suspect. If it's murder, they think they have the suspect. If it was suicide, then the person is not responsible for killing him and he took his own life. So we're going to see from the blood evidence if that's the case. So as you watch the video, write down the notes that support it being a suicide, and then if it's a homicide with someone being responsible. And I'm going to start